This is Dr. Rita Hijrasna, Assistant Professor at School of Life and Basic Sciences, Silas Campus, Jaipur. So first of all, I welcome you at the renowned University Jaipur, and I congratulate you that you are going to become a part of this renowned university. So today's in this session, I'm going to discuss with you a very important topic, that is enzyme, its properties and the factors affecting the enzymatic activity. So we know that in our body there are various biochemical reactions happens, and each and every biochemical reaction is require a particular kind of enzyme. Each and every reaction occurs in the presence of a very specific kind of enzyme. So enzymes are basically proteinaceous in nature. So most of the enzymes are proteins basically. Not all enzymes are proteins. There are some exceptions of the enzymes. They are not proteinaceous. There are some RNA catalytic molecules. They behave like the enzymes. So there are some exceptions like ribozymes, peptidyltransferases. These are the catalytic RNA molecules. They catalyze a reaction. So these enzymes are basically produced in a living cell, and they are highly specific and highly catalytic for their substrates. So highly specific for the substrate means each enzyme is very particular, very specific for the particular substrate. So a single enzyme will catalyze a reaction for a single substrate. So enzymes are a very important type of macromolecules. They are also known as biocatalysts because they are produced in the living cells, and due to their action, biochemical reaction in organisms carried out specifically and efficiently under mild conditions. So enzymes are basically made up of two type of parts, like proteinaceous part and non-proteinaceous part. So a holoenzyme, that is catalytically active enzyme, termed as holoenzyme, and its proteinaceous part is termed as apoenzyme, and non-proteinaceous part is known as cofactor. So this cofactor is essential for the enzymatic activity of a particular enzyme. These cofactors could be inorganic ions, or they could be organic compounds. Inorganic ions means like iron, copper, manganese, magnesium, potassium. These are the inorganic ions. They are essential for the catalytic activity of a specific kind of enzymes. And in case of organic compounds like TPP, thiamine pyrophosphate, FAD, NAD, and NADP and coenzyme A, these are the vitamin-derived organic compounds, and they are also essential for the catalytic activity of enzymes. So now I'm coming to the mechanism of enzyme. What kind of mechanism, how they catalyze the reaction. So I said that every enzyme is specific for a particular substrate. So each enzyme has a specific kind of active site or binding site for the substrate. A particular substrate will bind to the particular site on an enzyme, and then there would be interaction between enzyme and substrate, and they will form enzyme substrate complex. And then this substrate will convert to product, and the enzyme substrate complex is split into free enzyme and the product. So next is like, what are the properties of enzymes? So I said enzymes are highly specific for the substrate. They has high degree of specificity towards their substrate. Means this substrate specificity could be of various types, like substrate specificity, group specificity, bone specificity, optical specificity, geometrical specificity, and absolute specificity. So like group specificity, like each a particular kind of enzyme is highly specific for the group present over the substrate. Like I'm taking example of lactate dehydrogenase. Lactate dehydrogenase is basically used to convert. It is catalyze a reversible reaction where pyruvate converts into lactate. So this catalyze reversible reaction. So that is group specific, because in pyruvate and lactate they are same group. So in other kind of groups, similarity compounds also will catalyze by these lactate dehydrogenase. So there are various enzymes, like they will look at the specific group, that is why group specificity. And bond specificity and optical specificity. Optical specificity means ki, there are two optical isomers. So enzyme will specific to the L isomer or the D isomer. Geometrical specificity also, there could be a trans or cis conformations of a substrate. So the enzyme will specifically bind to either trans or either the cis configuration. Absolute specificity means a specific type of enzyme will bind to a single or absolutely a single that particular substrate, like urease enzyme. So urease will break down the urea into ammonia or CO2. So these are the different kind of specificity enzymes use towards their substrates. So second property, I am going to the colloidal nature of the proteins of these enzymes. So enzymes molecules are very large in size as compared to substrate, because enzyme has the cavity for the binding of substrate. So it is a very large as compared to substrate. So due to their colloidal nature, enzymes basically provide the large surface area for the interaction of substrate and enzymes. So that is provide the large surface area to interact with enzyme and substrate for the taking place of a reaction. And that is why the enzymes are required in a very small quantity to convert a huge amount of substrate molecules to products. And another thing is that enzymes are never utilized in a reaction. They can be reused again and again. So that is also a particular kind of property of enzymes. And enzymes are also very they has also a very high catalytic power. High catalytic power means their turnover number is very high. Catalytic power of an enzyme is measured by the turnover number. So turnover number is the number of substrate molecules converted into a product per unit time when an enzyme is fully saturated with the substrate. So in a unit time, how much substrate is converting to product? That is turnover number, and that is the measure of catalytic power. So if I'm talking about the mechanism of the enzyme, so enzymes basically do not change the equilibrium of the reaction, equilibrium of any kind of reaction. What they do, they basically decrease the activation energy of, or the energy barrier of a reaction, energy barrier that is present between the substrate and the products. So enzymes basically they provide a base to interact the substrate, so they basically increase the interactions rather than free molecules are freely moving in the solution without the enzymes and they convert to product. So that is why they decrease the activation energy or the energy barrier between the substrate and the product, and that is why more of the molecules can convert it to a substrate in a short time. So basically enzymes increase the rate of reaction by decreasing the activation energy of the reaction. So now I'm coming to the factors affecting the enzyme catalysis. So there are various factors they affect the enzymatic activity. So first I'm coming to the effect of substrate concentration. So substrate concentration means how much concentration substrate are in the solution to convert into product. So at lower substrate concentration, the active site on most of the enzyme molecules are not filled completely. There are various substrate binding sites are empty now. So there's no much more substrate in the solution that time, and so means since that point you are increasing the substrate concentration, the collision between the molecules would be increasing. That is why the rate of the reaction will be the rate of the reaction will increase according to the first order reaction. Means as the substrate concentration will increase, by that rate only the rate of the reaction only increase linearly. So in that case, you will get the linear uh, plot if we plot the graph substrate versus rate of reaction. So when the maximum velocity is reached, when all the substrate binding sites are occupied on the enzyme, so there is a limiting amount of enzyme. Initially, you add some substrate, then there's lot of binding sites are empty. When you increasing the substrate concentration, rate of reaction you will get the linear plot, and at the point you will get a point when all the binding site of enzyme will occupied by the substrate, and that time enzyme will be fully saturated with the substrate, and then if further you are increasing the substrate concentration, there will be no effect on the rate of the reaction, because there is no more substrate binding site are available for the substrate binding. So at that point, the saturation we will get. So increased substrate concentration after this point will no effect on the rate of the reaction. So therefore, the rate of the reaction increase as substrate concentration increase, but at the saturation level, you will see the zero order reaction. Zero order reaction means now the enzymatic activity is independence of substrate concentration. So in that case, we will get a hyperbolic plot. If we plot a graph versus substrate concentration versus rate of the reaction, then we will get a hyperbola plot. The next factor that affects the enzyme activity is the enzyme concentration. So if we have a vessel and there is no limitation of substrate in that solution, then if we will increase the enzyme concentration in the solution, then the rate of the reaction will be constantly increasing. In that case, we will get linear plot. But in other hand, if there is a limiting concentration of substrate in the solution, and then we are increasing the enzyme concentration in that solution. So initially, because enzyme is increasing, so initially there is a binding sites may be free, or maybe because there is a solution in the substrate in the solution, the substrate will going to bind to the increased concentration of enzyme. But at a certain time, when all the substrate will be occupied by the substrate binding sites on the enzyme, at that point, if we are going to increase the enzyme concentration, that will not affect again the rate of the reaction, because here there is no availability of more substrate. So there could be two possibilities, like you can get linear plot if there is a infinity amount of substrate or enough amount of substrate in your vessel, in your reaction, or if you are taking a limiting amount of substrate, in that case you will get hyperbolic plot. So the next factor, that is effect of product concentration. We talked about the substrate concentration, we talked about enzyme concentration. Now I am coming to the substrate concentration. So we know that the enzymatic activity, each and every reaction, metabolic reaction, they are highly regulated in our body. Because if there would be no regulation, then that is not possible to carry forward a proper pathways, metabolic pathways. So each and every reaction is highly controlled, under the highly controlled regulation. So enzymes basically inhibits by the various procedures, like here I'm talking about the product inhibition. So if there is a lot of product accumulated in your system, in that case this product will inhibit the first reaction of the pathway. So, or the first reaction of the enzyme, like there is a multiple steps in between the substrate or the products. Then, this if the product is accumulating, that product may inhibit the first enzymatic activity. This is known as feedback inhibition. So, if the product is accumulating, that will decline the enzymatic activity. Basically. So, next factor is the effect of temperature. So we know that enzymes are basically proteinaceous in nature, and proteins tertiary structure is basically highly sensitive towards the pH or towards the temperature. So each and every enzyme are basically highly specific for their optimum temperature. So if you are increasing the temperature will increase the kinetic energy of the molecules. So that is somehow unstabilize the protein molecule or the enzymes. So increase in the kinetic energy also increase the motion and therefore the frequency of this they collide. So collision will be high because kinetic energy molecules are getting the kinetic energy, and kinetic energy leads to the more collision between the molecules, and this basically affect the productivity or the enzymatic activity. So initially the rate of reaction with increase in the temperature to a maximum level, to the optimum level, will increase the rate of reaction. Suppose I'm coming from 4 degree to 30 degree or 37 degree, so or 40 degree. So initially when we increase the temperature from the 4 degree, at the 4 degree the collision interaction between the molecule is very less because less kinetic energy, less collision. So increase the temperature will increase the kinetic energy, will increase the collision, and that will basically increase the interaction between the molecules. So initially rate of reaction will be increase in temperature till the optimum temperature, and there will be a abruptly decline the rate of reaction, further increasing the temperature from the optimum temperature. So most animal enzymes separately becomes denatured at temperature above 40 degree Celsius. So till 40 degree, we will increase the temperature, the rate of the reaction will increase. After 40 degree, you will see a rapid or drastic decrease in the enzymatic activity. The next factor after temperature, I'm looking for the effect of the hydrogen ion concentration. Hydrogen ion concentration means the pH. So pH is basically minus log of hydrogen ion concentration. So hydrogen ion concentration plays an important role in our biological reactions. And the rate of the reaction is of any enzyme is drastically affected by the change in the pH, because every enzyme shows its maximum catalytic activity at a particular pH. Like digestive enzymes, like digestive enzymes, they are highly active at acidic pH. Some enzymes are active at alkaline pH. So pH is basically drastically affect the enzymatic activity. So most intercellular enzymes exhibit optimum activity pH between the 5 to 9. So if you are going above or below this range, the enzyme activity will be drastically changed. So in this case, we will get a bell-shaped graph in case of pH effect, because if you are going to suppose there is an enzyme that is highly active at pH 7. If you are going to acidic or you are going to alkaline nature, the catalytic activity of enzyme is equally disrupted. So in that case, we will get bell-shaped plot. Whereas in temperature effect, we didn't get the bell-shaped plot, because in case of temperature, initially till 40 degree temperature, the rate of reaction was increasing due to the increase in kinetic energy due to the collision of the molecules, but after 40 there is suddenly drastic decrease in the activity. So in case of pH, we are getting the bell-shaped graph, but not in case of temperature. So now I'm going to end my session. So thank you.